So we do have two speakers for today, the first of which is going to be Kathleen Gavin, who's an assistant professor in the Division of Geriatric Medicine. Kathleen first came to Colorado back in 2003 doing her master's at the University of Colorado Boulder. She went off to East Carolina University to do her PhD and then returned as a postdoctoral fellow working with Wendy Court and Dwight Clem in the Division of Geriatric Medicine. And she was appointed as an assistant professor in 2016. So Kathleen's work delves into what I think is a fascinating area, and that's the sort of the crosstalk and the developmental biology of how adipocytes arise. And she has a really lovely KO1 award that was awarded last year that delves into how bone marrow-derived progenitor cells actually contribute to the development of adipocytes, and in particular how gonadally derived hormones influence that and the differences between males and females. So that's what she's going to tell us about today. Kathleen?
Right, so thank you for the introduction and the opportunity to talk to you all today about my research. So let me get this started. So I'm going to be presenting a variety of information today, kind of a general background about the development of adipose tissue and why I think this is a really interesting topic. A variety of the information I'll be presenting is our data from my postdoctoral mentor, Dwight Clem, and then I'll transition to what my studies are today and how I would like to move them forward. So I just think adipose tissue is the most interesting organ, and one of the major reasons is because it has this extraordinary capacity to shrink or expand. So we know that there's people walking around with a wide variety of body fat distributions as well as total percent and total fat mass. So we usually discuss adipose tissue in the context of obesity and how we know that storing too much adipose tissue is associated with a variety of metabolic consequences. But we don't usually talk about is the other end of the spectrum, or people with lipodystrophy syndromes who are unable to store adipose tissue in the places that we usually are able to store it, so in depots underneath the skin and such. They instead have to store their excess nutrients in places like the muscle and the liver, so ectopic depots that actually lead to a variety of metabolic disorders that are very similar to what's observed in obesity. So what's really important here is that on both extremes of the spectrum, there's serious health consequences, centering us back to the idea that adipose tissue is critically important in maintaining health. So you need to be able to store at least some, and you need to be able to store it in the right places in order to maintain metabolic homeostasis and to maintain health. So the human body has a variety of depots in which it stores adipose tissue, and we're not going to get into the details of all of these depots, but what I do want to explain is that the influence of storing adipose tissue on health, a lot of it is determined by what depots you're actually storing that fat in. And so one of the common themes is storing adipose tissue in the abdominal region as opposed to your hips and thighs is actually more associated with increased disease risk as opposed to that more peripheral body fat distribution that could be benign or even protective in some cases. So the human has a variety of different adipose tissue depots, and these are important because adipose tissue is actually an endocrine organ. It's not just an inert energy storage site that mobilizes fuels when the body needs them, but it actually is an endocrine organ that secretes a variety of factors. A few of them are listed here on the left-hand side that actually go out and act on other organ systems to influence health. And some of these systems have a logical theme to them, so appetite and energy balance, insulin sensitivity, and glucose homeostasis. But other systems that the adipose tissue has an influence on are things such as angiogenesis, inflammation, and immunity. So different systems that you might not think to be associated with adipose tissue. And it's these adipokines or factors that are secreted from the adipose tissue that lead to those external influences. One other main thing that I think is really important to understand is that adipose tissue isn't just made up of fat cells. It's actually made up of a variety of different cells, and this diagram shows that slightly. So you can see here that in addition to these large unilocular cells, you have a variety of different cell types that are of a hematopoietic or myeloid lineage, so different lineage immune cells, and I don't think you can see this pointer, but what you can see is that there's a variety of different cells that accumulate in the fat tissue, and they comprise what is called the stromal vascular fraction of the adipose tissue. And in that non-adipocyte fraction, you also have blood vessels and neural innervation that impact the phenotype of that depot. So it's a combination of the fat cells themselves, the other types of cells that are in the adipose tissue that actually make up the phenotype of individual depots. So every depot has a different balance of these factors. We also think that within the adipose tissue, there are also different types of adipocytes. So we think that within one major white adipose depot, you might have a few different types of adipocytes that are also contributing to the phenotype, overall phenotype of that depot. So I want to focus for a few minutes on the three major types of adipocytes that are recognized to exist in human adipose tissue. So in the center here, we have the white adipocyte. So that's the major conventional type of fat cell that we think of when we think of adiposity. It has a large single lipid droplet, and its primary function is to store excess nutrients and mobilize them as needed. On the other hand, on the left-hand side here, we have a brown adipocyte. So we know that brown adipocytes do exist in adult humans now, and their primary function is actually thermogenesis or regulation of body temperature. And this is a type of cell that is actually trying to be harnessed to help regulate body weight in humans. So they get their brown color from their high mitochondrial content and high expression of uncoupling protein 1. So this uncouples the electron transport chain, allowing the generation of heat instead of the generation of ATP. So this is a really hot topic in the area of adipose tissue research. And on the other side, we have what's called a beige or a bright adipocyte. So this is kind of an in-betweener. So it's more like a white adipocyte and found in the same depots as white adipocytes, but it actually can be activated by the cold or adrenergic stimulation to increase its mitochondrial content and activity, increase UCP1 expression, and start to have a more thermogenic type of phenotype. So also people interested in different pharmacotherapies to activate the beige adipocytes in white adipose depots to try to help regulate weight. But that is not necessarily the focus of my talk today. Today I want to talk about how these cells are developed. So the dogma in adipocyte biology for years has been all new fat cells developed from adipose tissue resident mesenchymal lineage stem cells. So these are cells that reside and self-renew within the adipose tissue stroma, and when they receive the signals that they should commit to and become an adipocyte, they go down that pathway. So both beige and white adipocytes have long been believed to arise from a mesenchymal lineage that is now being worked out for details of how those precursor cells are actually different from one another. The brown adipocyte, on the other hand, still comes from a mesenchymal lineage, but it actually comes from a MIF5 positive mesenchymal cell as opposed to a MIF5 negative mesenchymal cell that leads to a conventional adipocyte. This is actually the same lineage that leads to muscle cell development. So showing that even within the recognized types of adipocytes humans have, there's a wide variety of different lineage pathways that these cells can come from. I'm here today to talk to you about an alternate lineage separate from myeloid cell or separate from the mesenchymal lineage completely, and to talk about how hematopoietic cells arising from a bone marrow origin, trafficking to the adipose tissue, and undergoing an adipose tissue transdifferentiation may contribute to development of a new type of fat cell. So we have here a CD45 positive cell that arises in the bone marrow, differentiates into a myeloid lineage cell that then traffics through the blood in its normal function, ends up in the adipose tissue. And we've known for years that myeloid lineage cells end up in the adipose tissue, but what wasn't thought is that it actually contributes to making new adipocytes. And we know that every year human adipose tissue turns over about 10% of its cells. So even if you're not gaining weight, you need to have a reservoir of cells in your adipose tissue ready to generate a new fat cell if the life cycle of a previous cell ends. So my postdoc mentor, Dwight Clem, was the first to discover these cells in adipose tissue of mice. And so he was able to conduct a number of studies determining that myeloid lineage cells transdifferentiate and become a marrow-derived adipocyte precursor, ultimately differentiating into a mature BMP, or bone marrow progenitor-derived adipocyte. Now, this is really interesting because from principal component analysis, these adipocytes are a very different cell population than their circulating myeloid origins, but they're also very distinct from white or brown adipocytes. So they do appear to be a new adipocyte of a new type of adipocyte in and of themselves, although we consider them most closely aligned with a conventional white adipocyte, so a single large lipid droplet, a low mitochondrial content. So Dwight's studies over the years have used really neat adaptive transfer models in mice. So bone marrow transplantation of bone marrow from animals who are tagged with a GFP, or in some cases have luciferase driven off of a promoter that's specific to the adipocyte. So he's able to perform transplantations and graft into a wild type animal, let that engraftment take over a period of weeks or months, and then look at either in vivo or take out the adipose tissue depots and measure the development of fat cells that have the tag from the bone marrow that was transplanted. So you can see in this image, this is a picture of digested adipocytes from a fat pad. So they're free floating, and you can see that there's a fair percentage of them that have this fluorescent green outline. And so this would be indicative of them coming from marrow that was transplanted into the mouse that was tagged with a GFP, or a green fluorescent protein. So we found in the mice anywhere from 5 to 25% of these cells can be coming from the marrow. And this is what we believe to be a physiologically relevant population of cells in the adipose tissue. So this is an example of some of the in vivo imaging that we can do with the luciferase tagged mice. So any of these little pixels here you can see would be luminescence that's produced from a cell that actually expresses that luciferase. And so we can measure in vivo accumulation over time of these cells after transplantation with bone marrow that has a luciferase being driven by, say, an adiponectin promoter, which is a very specific adipocyte gene. So we've measured the accumulation of these BMP-derived adipocytes in all the major fat depots, as well as some other organs that we know accumulate adipocytes in mice. So we found that in all the major fat depots, you can see in the blue bars that there is luciferase activity, which means there is evidence of production of bone marrow progenitor-derived adipocytes, but we don't find evidence of them in the muscle, liver, or lungs, so other organs we know to have adipocytes in them. One of the striking things that popped out at this information for us was that the gonadal and pericardial depots, which are two depots which are known to not be so healthy to store fat in, are two of the depots that accumulate the highest amount of these adipocytes. So this really interests us, saying maybe these adipocytes are accumulating or being produced more in depots that are more negative to health consequences. Dwight has also run some gene arrays on bone marrow-derived adipocytes in comparison to conventional adipocytes, and you can see that gene expression is very high for a large number of inflammatory type genes in bone marrow compared to conventional derived adipocytes. So the red color in this figure indicates really high gene expression, as opposed to the blue color in the conventional adipocytes being low expression of a wide variety of different inflammatory markers. And the very last one on the list there is leptin, and so that's a classic adipokine that we know adipocytes release leptin. And so in a conventional adipocyte, leptin level is very high, but we find in our bone marrow adipocytes, at least according to gene array, leptin is very surprisingly extremely low in these cells. We've also looked specifically at mitochondrial and peroxisomal genes and found that the bone marrow progenitor-derived adipocytes actually have a lower expression of these organelles that are known to be involved in energy generation. So white adipocytes have a higher expression of this panel of genes as opposed to bone marrow progenitor-derived adipocytes. So this, in some them accumulating in some of these more metabolically bad depots and having maybe a metabolically harmful phenotype, we think that there may be a story here with these cells, the production of these cells being linked to mechanisms underlying why certain depots are actually worse for your health than others. So let's just review the development of these cells and how we might try to measure them in humans. So this is just a different schematic that was published in one of Dwight's previous papers, demonstrating that the progenitors for these cells, we believe, are initiated in the bone marrow from these bone marrow transplantations that we've done. They start as hematopoietic progenitors, so CD45 positive cells that differentiate into a myeloid lineage cell. So in a mouse, this would be a CD11b positive cell. In a human, it's a CD14 positive cell. These cells traffic to the adipose tissue, where in the tissue, they undergo a transdifferentiation into a mesenchymal-like adipocyte progenitor. And at this point, they lose those surface proteins that tell their story about where they came from. So if we just look at them in a human adipose tissue, by the time they become committed to the adipocyte lineage, they actually don't have those markers to tell us where they came from. So in an animal, it's easy to continue studying these cells because they have their fluorescent or GFP tag that we gave them for their bone marrow transplant. But in humans, we hit this roadblock. We don't know how to study these in humans when we can't do these fancy lineage tracing studies. What we were able to come up with was a collaboration with Dr. Jonathan Gutman in the bone marrow transplantation clinic here at UCH. And he was able to allow us access to some of his patients who had had a bone marrow or hematopoietic stem cell transplant from another donor, so an allogenic transplant, who were doing well, thriving after their transplant. They came into our lab and we obtained an adipose tissue biopsy sample from them. So from that adipose tissue biopsy sample, from just next to the belly button, we were able to isolate out a very pure population of fat cells. And in that cell, we analyzed the DNA of the cell. And we looked for specific short tandem repeats that identified either the donor from the transplant or the recipient of the transplant. And if we were able to measure donor DNA in those fat cells, that was indicative that those cells were part of the cells that were transplanted as part of that clinical procedure. So we brought in about 8 patients who were anywhere from a year to about 3.5 years after their transplant, and we were able to find consistently in 7 out of 8 patients that about 5 to 35% of their adipocytes had evidence of donor DNA. And we could actually see, if you see some of these dots are colored, so 2 of them are blue, 2 are red, and 2 are green, those are repeat biopsies. So we had patients come in and take a biopsy about 6 months to a year after the first one, and we're able to see an increase over time. So longer duration since transplant, we found more evidence of donor DNA. So this was our first evidence that these cells do appear to exist in humans. Now, unbeknownst to us, there was a group in Sweden that was conducting an identical study. So a much larger study. Their healthcare system allows them access to, they had about 65 patients in their study, but Dwight's basic research had stimulated them to want to look at this in humans and see the translational relevance. So they had about 65 patients and they found basically the identical results to us. So about 5 to 40% of adipocytes of their patients were found to be of donor DNA. And they had enough patients in their study to actually stratify them into different categories according to BMI. So they had lean, overweight, obese, and severely obese patients, and they found that actually more cells were produced in the obese patients from the bone marrow-derived lineage than those of lean patients. So again, we think obesity and fat mass is playing a role in the production of these cells. So because so far the only way we have to measure these cells in humans is from patients who've had a bone marrow transplantation, I've been trying to move forward in measuring these cells in humans, but without a novel protein biomarker to identify these cells once they're a mature adipocyte. I've had to be creative in how we go about studying them. We're on the hunt for a novel protein biomarker, and I'll get to what we're doing for that. But for my K award, what I've proposed to do is I've been developing a primary cell culture model so that I can isolate these cells out of the adipose tissue when they're still at the progenitor stage, where they still have their surface markers saying what their lineage is, put them into culture, and put them through a process where I can differentiate them into adipocytes. So I'm not going to get into the details of the methodology, but I use a stringent flow cytometry sorting strategy to isolate out conventional lineage adipocytes, which are CD45 negative on the left-hand side, as opposed to CD45 positive or hematopoietic lineage cells, and we can separate that into myeloid lineage cells, so macrophage, monocytes, neutrophils, and then also a lymphocyte lineage. So all of these cells are coming from human adipose tissue biopsies we bring for patients who come in specifically for research studies. We isolate out the adipose stroma and are able to sort these cells in the flow cytometry core. So then I propagate and differentiate them in culture. So we grow them to confluency, put special differentiation media, and they take up lipid. And we know from some proteomics analysis that they do indeed express the major adipogenic proteins or adipocyte proteins we would expect. So these are turning into bona fide adipocytes and not just myeloid cells accumulating lipids. So I'm culturing up these cells to compare conventional lineage white adipocytes to bone marrow progenitor-derived adipocytes that are differentiated in culture. So I'm doing studies on differences in their proliferation and differentiation. We're performing proteomics. And so this is the technique we're using at this point to try to determine a novel protein biomarker for these cells. So is there a candidate surface marker we could use an antibody, run these cells through flow, and say, 20% of these cells came from a bone marrow origin. And so we're hunting down something so that we can bring in anyone off the street, take an adipose tissue biopsy, analyze their cells, and know what percentage of their cells comes from the bone marrow. We're also doing in vitro metabolic phenotyping and metabolomics. So I've shown you some gene array we've done in the mouse cells, but we really need to hone in on what is the metabolic phenotype of these cells from humans. Are they harmful potentially? Do they have more? Are they more insulin resistant? Do they have lower mitochondrial activity? Things like that. And then I'm just going to end with a couple factors that I'm really interested in that we have found, at least in our animal studies, that influence the production of BMP-derived adipocytes. So one is male versus female sex, and the other one is ovarian hormone status. So when Dwight started doing these studies, he found consistently that the female animals had greater production of the BMP-derived adipocytes as compared to the male animals. So you can see this with the in vivo luciferase imaging, the female having more light emission. We also found that consistently, again, in both animals and the major fat depots, we see luminescence, but we see a significantly lower amount in the male adipose tissue from the male, specifically in the gonadal region, although a tendency in the perirenal and subcutaneous as well. So that got us thinking, what is the influence of sex hormone status in these animals? I'm in the division of geriatrics. I'm really interested in aging in men and women and how body composition and disease risk changes with age. So we were interested in setting up a study to look at how removing the ovaries of animals might impact the accumulation of these cells. So we compared overrectomized mice to control mice and found that indeed, as you can see here, increased light emission meant increased production of BMP-derived adipocytes. And we also used a whole body ER-alpha knockout model to see if we could determine if it was signaling through ER-alpha that might be playing a role in this. And we see similarly to the OVX animals, estrogen receptor alpha animals do have an increased production of these cells. When we take out the adipose tissue depots of these animals, you can see, again, the gonadal depot consistently has a higher amount of production of these cells. Compared to wild type in the black bar, overectomy in the red and ER-alpha knockout in the purple have a significant increased production in each of these gonadal and subcutaneous depots. When we added back estrogen to a subset of the animals, it actually returned them to wild type or even below the production of BMP-derived adipocytes. But when we replaced with progesterone, we didn't see that same effect. So with the OVX plus estradiol add back and the ER-alpha knockout, we think we're getting down the road of estrogen signaling through ER-alpha being important in regulating production of these cells, at least in female animals. So my K award is trying to get in more depth in a cell culture model, exposing these cells to the sex hormone environment that they would see in vivo and trying to understand how this is impacting the production and differentiation of these cells. So overall, adipose tissue is critical in maintaining homeostasis. And we now know that bone marrow progenitors may be an important contributor to adipogenesis or generation of new fat cells. Production of BMP-derived adipocytes may be a mechanism by which excess adiposity increases disease risk. So we have a long way to go to actually prove this hypothesis, but I really think that so far our preliminary data and our publications to date really support this. And I think that understanding adipocyte lineage, whether it be cells of the BMP-derived lineage or brown or beige or other fat cells that might actually be in the adipose tissue that we don't know about yet, knowing more about them will help improve our development of strategies and interventions to help improve health and prevent accelerated chronic disease risk, particularly with aging. So just want to thank my mentors, Dr. Clem and Court. This has been not an easy area to study and had some mixed reception in the adipocyte biology world, and so we've had to work really hard for people to start accepting this, but our paper in humans, along with the group from Karolinska Institute, has really solidified this as an important area to research moving forward. So thank you. I'll take any questions. Yeah. Yeah. Yes. So that's a good question. So I don't know offhand the percentage of increase. I know the overectomized animals had a significant increase in whole body weight and on the depot level. We didn't see the same unexpectedly. Our ER alpha animals didn't gain as much weight as we expected to, although these animals were still really pretty young. But the estrogen add back brought the whole body weight as well as the depot weights back down to wild type. So we did see this reflected in body weight at a whole level. I think we're still working on getting the exact body composition information, but it did influence that.
So can you distinguish?
Yeah, so that's a really good question. A lot of, the field is now beginning to accept that these adipocytes probably are, or they are real. Whether they're real outside the context of insults and medications from a transplantation is uncertain, at least in humans, because we haven't had any specific evidence, or we don't have a way to measure them yet. However, Dwight has used some models that are not bone marrow transplant models. So he has a Lyce-M-Cree model that he uses, and forgive me because I'm not a basic scientist or an animal researcher and haven't done any of these studies, but I know he has used at least one model that does not require a transplantation and is able to see the development of these cells. But I think being able to find a novel protein biomarker, measure these in a patient who hasn't had a bone marrow transplant is going to be critical in making sure that the knowledge moves forward that these are actually an important population of cells in a healthy human that hasn't had extensive therapy or medication. But I'm really interested in what the signals are for mobilization, or even within the adipose tissue, for this unconventional transdifferentiation. So is there something in the microenvironment of the adipose tissue, some signal telling more of these cells to end up in the adipose tissue? So there's a lot of questions that we still need to answer. Yeah. So what would mobilize, as far as you mean like mobilizing the lipids that are stored in the adipose tissue for fuel sources? So things that would be like things that generate lipolysis and things like that? Is that what you're talking about? So that would be things where if the body is fasting for a long period of time and you have decrease in insulin. Yeah. So, it is a good question if when we need to mobilize nutrients or energy from adipose tissue, if these cells are resistant to that, where maybe a conventional adipocyte would release more lipids. So are these cells more insulin resistant? And that's one of the ways by which they have a negative metabolic phenotype. So we're uncertain if if there's different composition of adipocytes within an adipose tissue depot, if that depot receives signals that nutrients need to be released, if there would be a strategy for the adipose tissue to be releasing from some types of cells and not others. So we have a lot of metabolic phenotyping to understand if there are true differences in some of the adipose tissue's main functions, which is releasing nutrients as the body needs them. Is that process different in this type of adipocyte than a conventional one? And I would hypothesize that it is, that they have different sensitivities to some of these factors, but I need to conduct the tests to know that for sure. So I'm sure that's, there are groups doing adipocyte biology that have done some RNA-seq in different populations of cells. That's something I've been discussing recently with Paul McLean and his group, and the advantages of using RNA-seq to interrogate what's going on in some of these cell populations. So Dwight's used gene arrays, and now I'm trying to go into some of these new areas of proteomics, metabolomics, RNA-seq to see if we can really hone in on really what's going on at the cellular level. Yeah. Yes. Yeah, so I think in beyond just I plan to study conventional as compared to myeloid lineage, but the brown and the beige, there's a whole spectrum, and now there's evidence that endothelial progenitors also develop into fat cells, and are those the same as these other types? So I think there's a wide variety of, it's a matter of being able to separate out these different cell types and do studies on the individual cell types instead of a tissue homogenate.